Thank you everyone for joining. We are very happy to have Marco Saltini today. So Marco is working with Bella Mulder, and we'll talk about microtubule today, so a subject that is dear to my heart. So Marco has been working on microtubule for quite a while already, and so with some study on the regulators, important regulators of microtubule, and maybe a little bit with a focus on microtubule stability. I don't know if you would agree with this. And so actually, so the work that Marco is going to talk about today, so I'm not, of course, not going to talk about it, but I think it's very interesting in the sense that it brings experimental data, like experimental observation and computational modeling to understand the emerging property of microtubule, which is really typically a central focus of what we do in this journal. So, and of course, with a very important implication for developmental biology. So we are very happy to have you here today, Marco, and the screen is yours.
Thank you very much, Olivier, for the introduction, and of course also for inviting me here, because it is indeed very nice to have the chance to present this paper to all of you and to all the readers of Quantitative Plant Biology. So, okay, so this work builds on basically on a series of developments that we have achieved in the last 4 to 5 years, and it is mainly a theoretical paper, but it is based on, as you said, as Olivier just said, on experimental data that we have collected and our collaborators have collected in the last few years. And it concerns a very specific mechanism in a plant cell, which is the longitudinal lock-in of the cortical microtubule array after reorientation, after exposure to blue light. But let's start from the beginning, which means that I will give a very quick introduction about this system. And as you of course all know, the cortical microtubule array is a very highly structured, it's a highly ordered structure in plant cells, and it is responsible for many cellular processes and needs to drive the growth and the morphogenesis of the cell, and therefore also of the organism of the plant as a whole. Its function is strictly connected to its spatial organization, which depends on the dynamics of the microtubules that are part of this cortical array. And in particular, central to the spatial organization of the cortical array is the generation of new polymers. In plant, as you all know, polymers like microtubules are generated in 2 distinct ways, which means that can be either generated through a nucleation template, for instance, a gamma tubulin complex, which in the case of plant microtubules is most of the time located at the microtubule lattice, or through severing events of already existing microtubule. Katanin has been identified as one of these proteins that can that can sever microtubules, and by severing one microtubule, we basically end up with having one more microtubule. So it is a process that results in the effective generation of a new polymer. Now, this second mode of nucleation, of generation of a new microtubule, is has been revealed to be responsible for the rapid reorientation of the cortical microtubule array. Here we can see the image of the cortical array of a dark-grown hypocotyl cells of Arabidopsis thaliana. And what we can observe is that starting from an initial orientation of the microtubule array, which is transferred with respect to the growth direction of, maybe we can see it again, transferred with respect to the growth direction of the cell, we have a sudden reorientation from this array from a transverse direction to a direction which is longitudinal to the growth direction of the cell. Now, this orientation needs not to be intended in a purely geometrical sense, in the sense that this orientation consists of the depolymerization of the disappearance of the initial array, which is replaced by a new array in the opposite direction, in the longitudinal one. Now, the generation of all these polymers in the longitudinal direction, it is due to the effect of Katanin. In particular, Katanin has been shown that it localizes at the crossovers between microtubules with different orientation. In this case, it localizes at the crossovers between a transverse microtubule and the longitudinal microtubule. and preferentially severs the newer microtubule between the two. In this case, of course, since we start from a transverse array, new microtubules are those that are oriented in the longitudinal direction. And the iteration of these kind of severing events results in the effective creation of this new array in the longitudinal direction. Now, in previous works, we have identified what are the main drivers of this reorientation, which means that we identified what are the factors that play a major role in this reorientation in order for the reorientation to occur. In particular, what we have observed using like a combination of experiments, computer simulations, but also analytical theory, is that the preferential severing of longitudinal microtubules, it is very important for this reorientation to occur, as well as a very high microtubule end stability. To be more precise, we have identified two proteins, which are spiral 2 protein and clasp protein, that are indeed, of course, potent stabilizers of respectively microtubule minus end and microtubule plus end. And we have observed that in absence of these proteins, it is impossible, or at least, at the very least, it is very, very hard to obtain a reorientation, an amplification in the number of longitudinal microtubules. Now, an important thing of these previous works is that they focused only on the initial stage of this reorientation mechanism. Indeed, if we think about like what can happen for at a longer time scales, we can argue that once the new array, or better, once the array has completely reoriented and it is directed in the longitudinal direction, we can have exactly the same mechanism that occurs in the opposite way, which means that we start from longitudinal, from the cortical array that it is oriented longitudinally with respect to the growth axis of the cell, and the occasional occurrence of nucleation in the transverse direction will, in principle, be able to amplify, in this case, the number of microtubules in the transverse direction and then go back to the previous state. But actually, this is not what it has been observed by the experiments. Indeed, the experiments or experimental observations have observed that this new longitudinal direction for the microtubule, for the cortical microtubule array, is maintained over long time scales. So the question is now, okay, how it is possible that we can keep this array oriented in the longitudinal direction? Why don't we have then a redistribution of tubulin between these two arrays, such that eventually we end up in a, for example, in an isotropic condition in which we have microtubules oriented in all the direction? Or why don't we have cycles between longitudinal polymerization and transverse polymerization? And this is pretty much the question that we want to address. So how is the reoriented array maintained in the new longitudinal direction for long time scales? To address this question, we set up a stochastic model of microtubules undergoing dynamic instability in a finite pool of tubulin. In particular, here, we want to study the competition between two classes of microtubules, longitudinal microtubules and transverse microtubules, when they compete for the same share of resources, which is, you can think about it as a pool of free tubulin. It is very important to understand that in our model, we wanted to, let's say, to make the model as simple as possible in order to have a good mathematical explanation of what happens. And in this sense, basically, our two populations of microtubules, they do not really interact with each other, apart from the fact that they share the same pool of resources. Indeed, we do not take really into consideration the cell surface. We do not take into consideration the actual geometry of the cell. We only want to make a model in which we have competition between two different populations. And we want to see if the asymmetry induced by, for instance, the preferential severing for the longitudinal microtubules is enough to explain this lock-in the longitudinal direction of the microtubules of the cortical array. But let's try to go through the model a bit more into the details. As I said before, the two populations of microtubules compete for the same pool of tubulin, which means that they receive building material by the pool of free tubulin and basically they undergo dynamic instability. So they grow with a growth velocity, with a growth speed, which is dependent on the amount of free tubulin. And when they shrink, they release building material back to the tubulin pool. Not only the growth speed of microtubules is dependent on the amount of free tubulin, but also the nucleation rate of new microtubules. Here, we try to make it, again, as simple as possible, just by saying that there are two possible modes of nucleation of new microtubules. So either from a gamma tubulin complex, which is located in a dispersed part of the cytoskeleton, which means that it is not attached to any microtubule, or new microtubules can be enucleated from a gamma tubulin complex, which is located to the lattice of an already existing microtubule. In such case, we can have, in our model, we assume that there are two possible directions for the nucleation of the new microtubule. So either the new microtubule is nucleated parallel to the mother microtubule. So basically, a microtubule creates a new one in the same population. So longitudinals create microtubules in the longitudinal direction. Or it is also possible that a microtubule-based nucleation creates a microtubule for the opposite population. So it gives an advantage to the competing population. Now, in our model, we make the strong assumption that this mode of nucleation is the most prominent one. So most of the newly nucleated microtubules are nucleated in the same direction, or if you prefer, in the same population as the mother microtubule. Finally, also our microtubules, as I said before, undergo dynamic instability. And to model dynamic instability, we basically make an extension of probably the most used model of microtubule dynamics, which is the Docter and Leibler model, in which microtubules spend periods of time in the growing state, and they can undergo a catastrophe, they can shrink, they can recover their length through a rescue, and so on. Apart from that, as I said before, we also include the possibility for a microtubule to undergo a severing event. Now, after a microtubule is severed, the newly created plus end can grow with a certain probability, P+, or it can shrink with a certain probability, 1 minus P+. After this happens, of course, the newly created microtubule is still subject to dynamic instability. So it can, like, for example, a shrinking microtubule can still recover the initial length through a rescue, or this one can also undergo a catastrophe and shrink and depolymerizes completely. Apart from that, the very last point of the model is the severing. As I said in the introduction, what we can observe from the in vivo observations is that usually when there is a crossover between a transverse and a longitudinal microtubule and a severing event takes place, the longitudinal microtubule is severed with it's more likely severed rather than the transverse microtubule. Here, however, as I said before, we do not really take into account a real interaction between the two populations of microtubules. But in the same way, at the same time, we want to include this preferential severing also in our model. And we include this preferential severing in our model just by assuming that the intrinsic severing rate of a single microtubule is higher in the case of the longitudinal microtubules compared to what happens for the transverse microtubules. And we basically call this preferential severing Q. Q is basically the fraction. You can see it as the fraction of microtubules that are severed in the longitudinal direction compared to all the severing events that can occur. So if Q is higher than 1/2, than 0.5, we have that the rate of severing for longitudinal microtubules is higher than the rate of severing for transverse microtubule. So the model is, in a nutshell, the model is this. what we did then, it was try to run stochastic simulation to observe if this asymmetry induced by this bias in the severing of microtubules belonging to two different populations is enough to explain the long-term lock-in of the longitudinal array. But what we observe is that no, it is not enough. If we just assume that the only asymmetry in the system is given by the preferential severing, we do not observe reorientation at all. Even in the case in which we study our model using unrealistic numbers, using unrealistic parameter, which is in this case given by this probability of stabilization after severing for a newly created plus end, even in this case, that we can observe some kind of degree of reorientation of microtubules as a function of time. We can indeed, at the end, we can see that there are more longitudinal microtubules than transverse. The number of transverse microtubules is still pretty high compared to what we saw in the video. What we really want to see is that we have a very strong depletion of microtubules in the transverse direction. And we also want to see that we have a strong depletion of the tubulin used by the transverse population, which is not something that we see here. Here we still see that the tubulin used by the transverse population is still rather high. So there must be something else. There must be some other kind of asymmetry in the system such that this lock-in, this longitudinal lock-in can occur. Now, for that, probably we need to take a little step back. It is now accepted the fact that cell geometry can influence the dynamics of microtubule. Indeed, it was a work from 2008 from our host Olivier Amann, who showed that microtubules in cells tend to align along the lines of maximal stress. At the same time, a more recent computational study from Banda and Chakraborty have revealed that as soon as you add little cues to microtubule dynamics in one direction or in another, like if you add little asymmetries, which can be, for example, dependent on catastrophe induced at sharp cell edges, we can select a preferential alignment for the cortical array with respect to another alignment. So based on these premises, our idea is that since there must be another asymmetry between the longitudinal and the transverse population, we assume that this asymmetry, it is in the dynamic parameters, it is in the dynamics of the two different populations. In particular, what we say, or what we hypothesize is that longitudinal microtubules grow slightly faster than transverse microtubules. And it doesn't need to be very, very faster, like much faster than the transverse one. Just need to be slightly faster than them. And we want to see if an asymmetry in this dynamic parameter is enough to create this longitudinal lock-in, which is now our hypothesis. Can a small orientation-dependent bias in the dynamics, can that cause the longitudinal lock-in? Here, as I said before, we studied the case in which we have a small advantage for the growth speed of longitudinal microtubules, but we can very well assume that these imbalances in the catastrophe rate, maybe longitudinal microtubules undergo catastrophes less frequently than transverse microtubules. The final outcome of the model, it is exactly the same. Now, if we run stochastic simulations again on this model, what we observe here is that even with a small bias in the growth velocity, here it is like 10% for the longitudinal microtubules, is enough to make this full reorientation occur and be maintained by the cell. Here, indeed, you can see that number of longitudinal microtubules is much higher than the number of transverse microtubules at the end of the process. In the same way, we can see that the tubulin used by the longitudinal population is much higher than the tubulin used by the transverse population. So it looks like that even by adding a very small advantage for one population over the other, we can observe a dramatic shift in microtubule orientation. And it is probably even more evident in case we play a little bit with the parameters. Now, let me guide you through these heat maps in a very, let's try to do it in a very simple way. These heat maps represent how efficient and how fast the reorientation is when we change the parameter P+, which was the stabilization, the probability of stabilization after severing of newly created plus ends, and when we change Q, which was the preferential severing for longitudinal microtubules. In the top row, we have this efficiency and speed of reorientation in the case in which we have this bias in the speed. Here in the bottom, we have basically the same efficiency or speed of reorientation in case we do not have any speed bias. Lighter colors correspond to a more efficient and faster reorientation. And you can see indeed that here in the top row, colors are much lighter than here, which means that we have more room or more possibilities of a combination between the parameters of the models, such that in the case in which we have a bias in the speed, we can observe a full reorientation and a fully maintained reorientation. So now one can, of course, argue, okay, maybe all these things, they sum up together, and you can have this you can have the lock-in, the longitudinal lock-in, only if you have a bias in the speed, if you have a high probability of stabilization after severing, and also if you have a good degree of preferential severing for the longitudinal microtubules. So now to see if that is indeed the case, we study analytically the model in which we do not consider any possible severing event. So we study the model in which we only have the two populations that compete for the finite pool of tubulin, without considering any kind of interaction between the two populations, without considering any kind of severing event. But even in this case, what we can observe is that longitudinal microtubules eventually end up being many more than transverse microtubules once the equilibrium is reached. Now, this plot you can read this plot in this way. On this axis, we put the propensity length for dispersed nucleation, which means that the more we are close to zero, the more likely is that a new microtubule is nucleated through a microtubule-based nucleation, which means close to zero, many more microtubules are nucleated parallel to the mother microtubule in the same population of the mother microtubule. And here on this axis, we put the final number of microtubules once the process is finished. So here, in case in which we have a small advantage for the longitudinal microtubules, and in case we do not have any severing event, what we can observe indeed is that even in this case, in case we have a strong bias towards microtubule-based nucleation, the reorientation and the lock-in still occurs, and it is still very relevant. So one indeed can eventually argue that severing events, in fact, are not important for this process to occur, but we will get there later. But now, let me now try to sum up a little bit. First of all, the first important point is that tubulin, like scarcity of tubulin, is very important for this mechanism to occur. Indeed, in a previous work, we studied this system in case tubulin was available in abundance, basically until the end of the process. In this case, what we could observe is that the number of microtubule exponentially grows and grows and grows without ever ending. So this model, after a short time, basically loses any kind of biological significance. So we cannot really observe reorientation of the cortical array in case we only we think about a system in which we have as much tubulin as we want. So like, long story short, tubulin needs to be, or better, dynamics needs to be constrained by scarcity of tubulin in order for this mechanism to take place. The second point is that a small advantage for one population of microtubules over the other with a small advantage, we can obtain the final array orientation. In the model, in the model that I showed you before, we assume that this small advantage was in the growing speed of microtubules. But at the same time, we can assume that this small advantage was could have been in the intrinsic nucleation rate of longitudinal microtubules over transverse one, or in a slightly reduced catastrophe rate. Of course, I didn't show you this in these slides, but assuming this, making this assumption, basically make us end up exactly with the same result. We can observe no difference whatsoever. And this is actually a very nice example of what in community dynamics community ecology is called the competitive exclusion principle, which basically states that when you have two populations, any kind of population that compete for shared resources, the one that have even the slightest advantage over the other prevails, while the second one, the one who has the disadvantage, is bound to go extinct. Now, the fact that we do not observe complete extinction of transverse microtubules might be due to the fact that our model can still include some random generation of microtubules in the transverse direction. But it is very interesting to understand that this mechanism can only work in case in which we have a strong bias towards the nucleation of new microtubules that are parallel to the mother microtubule, which is exactly what happens in microtubules from the cortical array. So finally, as I said before, one can argue that preferential severing and stabilization after severing are not really important for this reorientation mechanism. However, taking a look at our data, we can observe that while on the one hand, it is true that they are not strictly necessary for the reorientation to occur, but without that, the reorientation may take a very, very long time. So preferential severing and stabilization after severing are still very important, factors for this reorientation to occur in a fast way. Now, this is pretty much it, and of course, due to our result, we can of course try to argue what can this asymmetry be, because of course we hypothesize that an asymmetry in the dynamics between the two populations is plausible, but for what we know at the moment, this asymmetry, it has not been observed quite yet. So can the asymmetry be experimentally observed? Well, in principle, yes, because if we make a thorough analysis of these videos of microtubules in the cortical array that undergo dynamic instability, and we have a good amount of data, we can in principle compare the dynamic properties of microtubules in one direction and with the dynamic properties of microtubules in the other direction. And if we have enough data, we may be able to detect even a small advantage for one of the two, which according to our model would be enough to explain this reorientation and the persistence of the new orientation of the array. Then, of course, if this dynamic asymmetry is detected, one can always wonder what can a molecular mechanism be such that this asymmetry exists. Unfortunately, we do not really have an explanation for that. So as I said before, we think that it is plausible to have this asymmetry, but we cannot really think about a molecular mechanism that enables this asymmetry. What can be? Because honestly speaking, if we think about the what we know at the moment is that, for example, for cells which have pretty much a cylindrical shape, like for example the cells close to the root, to the elongation part of the root, what we can observe is that microtubules tend to align in the transverse direction rather than in the longitudinal direction because of stress, because of stress. So they tend to align over the lines of maximal stress, which is the transverse direction. So for the moment, we cannot really find a good explanation for this possible asymmetry. Finally, the model was really highly idealized, which means that, as I said before, we wanted to build a model that was handleable and tractable from the mathematical point of view in order to find what were the key parameters that enabled this longitudinal lock-in. Of course, now the next step is to try to find a more realistic model for cortical array dynamics. Then, of course, it is not possible to study this process from a mathematical point of view, but for sure we can do it computationally. And interesting enough, a framework such that includes some more, such that like all the possible repertoire of interaction between microtubules, such that, for example, real crossovers between microtubules or catastrophes induced by cell shape, or, for example, zippering, like a framework, a computational framework that includes all these possible interaction actually already exists. So we can just take this framework and add this higher degree of complexity, which is a bias in one of the, for example, in one of the dynamic parameters in one direction, and see if this more realistic model can confirm our finding in our toy model. And with that, I would like to conclude. And first of all, I would like to thank my co-author and former supervisor, Bela. And of course, also Yelmer, Masayoshi, and David, who are the experimentalists who performed all the experiments our model was based on. Because, of course, without their work, this paper would have never been written. And of course, I would also like to thank you for listening.
Thank you very much, Marco. It was inspiring and very clear. So maybe I'll start with with a few questions. I'm going to abuse my status as a host, I think. So yeah, maybe on the, for instance, the Katanin. So I maybe to start to digging with the molecular mechanism, because I agree, it's a no. But since there's some reports showing that Katanin is targeting preferentially errors in the microtubule lattice. How would you see this in your model? Would you expect to have more errors in the longer microtubule, for instance?
Well, that might be a possibility. Indeed, of course, microtubules in the already existing microtubules are older, so it might be that defects are more frequent than in the case in which we have newer microtubules. An alternative hypothesis might be that it is due to the way that they stack on each other, because newer microtubules, they tend to lay on top of the older ones. So it might also be that Katanin targets longitudinal microtubules or newer microtubules more than older, just because they're on top of the other. But okay, for the moment, I cannot really give a clear answer to that. We are still.
Yeah, but I was thinking that maybe that's something you could explore with your current model, like with the time.
Oh yeah, that's. I mean, by adding, we can add another degree of complexity by saying that microtubules are severed more often in case they are old in some specific places on their lattice due to the if that microtubule was at that length long enough such that a defect might arise. Yeah, that can be done for sure.
That's a follow-up again. Yeah. I see that there's a question, so I'll go on with my discussion afterwards. So from Mateusz Majda, actually not too far from, in the journalist, I was wondering what could be the molecular mechanism behind the cell geometry-induced alignment of microtubule, and how the polarized cell shape could appear before microtubule alignment. So I guess
well, okay.
Large question.
I'd say that we are, of course, in the field of speculations, because, as I said before, at present we never really observed any difference in the dynamics. But might also very well be that in the moment in which you beam the cell with blue light, might activate some downstream effect and some proteins that can override the typical transverse alignment. I don't know, I'm just guessing that maybe some stabilizers they detach from the transfer microtubule to attach to longitudinal microtubules just due to a downstream effect, which is due to blue light. But that is a pure speculation for the moment.
Yeah, no, it's open. Yeah, I have a few more, but I see there's more questions coming. So from Yushen Long, Singapore, thank you, Marco, for a nice talk. I recall some recent theoretical and experimental data suggesting that longitudinal alignment is a default, whereas transverse would require extra bias. Could we interpret your model by considering such removal of bias compared to the nucleation growth bias?
Okay, I first of all admit that I'm not aware of this theoretical and experimental data. So indeed,
well, I can fill you in on this one. So it's I mean the idea is that if you have an elongated cell, the flattest part of the cell is along the long axis of the cell, and it's curvy on the transverse. So if you take into account the persistent length of the microtubule, you would think that the microtubule would be more stable along the. So I guess that's what Long has in mind.
Well, that's in that sense, yes. But again, as far as I know, what microtubules tend to do usually is to, indeed, of course, you know that much better than me. They tend to align over lines of maximal stress, which in the case of a cylindrical cell, it is the transverse or the lateral surface of the cell. Then, I mean, of course, we can, since we are in the field of theory, we can definitely make a model such that the dynamics, or for example, the polymerization of microtubules is faster in the longitudinal direction due to like less constraint due to the geometry. That for sure that can be done. But
so you could. So I guess to follow up on this one. So I was thinking that so in one hypothesis we have with the stress response, the microtubule response to stress, is that they grow faster with maximal tensile stress. So you could imagine that when you put blue light, it's actually the stress level that is decreasing. And so, for instance, you drop in turgor pressure or something, I mean, the growth is stopping. And so you could, so I guess what maybe Long is saying as well is that you could have a bias in the transverse direction when the cell is turgid in the dark. And then you remove this bias, and then you unleash another bias in the longitudinal direction.
Yeah, that might be. Yeah, that might very well be. Indeed, probably it's a good idea to explore this possibility, because it can indeed create, as you said, this light advantage for the longitudinal population over the transverse one. But I think that first of all, we need to find this asymmetry, because at the moment we are
for sure
still in the phase in which this asymmetry is only hypothesized but not observed yet.
Yeah, I completely agree. Yeah, we need more experimental support.
But I think it is a good idea. Yeah. Indeed.
But I guess maybe in the model, because here it's an absolute bias, right? So it's but I don't know if it would work as a relative bias, like as a dynamic bias. So like losing a bias in the transverse direction, would that be enough to to induce a long-term stability of the microtubule in the longitudinal direction?
What do you mean with losing a bias? Because
like as if well, maybe it's a bit too specific of a discussion now. So if the microtubule were growing faster in the transverse direction, and then you put blue light, and they grow equally at the same speed everywhere, but now there is a relative difference, right? So would that be enough to switch to a longitudinal probe?
I don't think so, because it would be basically exactly this model, in which in this case we start from a configuration which is more longitudinal microtubules, less transverse microtubules. We just change the labels, so everything that had the parallel sign becomes orthogonal side, and vice versa. If we do that, basically we end up exactly as it is stated in the at least in the model in which we didn't have any difference apart from the preferential severing. Which, yeah, so this model is pretty robust upon changing of the initial conditions. So I don't think that would help.
Fair enough. Okay, I'm looking if there's more questions. So maybe I have another one. There was this because there's this idea that when the crowding can have an effect on the microtubule dynamics. Mm-hmm. And so, well, actually, maybe it comes down to more speculation, actually. But you could imagine that when you shine blue light, for some reason, you affect the crowding of the cytoplasm, or as I said, I mean, maybe it's Tucker pressure. And there was this idea that this would slow down microtubule dynamics. So in the hyperosmotic condition, you would slow down the microtubule dynamics. So I was wondering whether you think this could be still asymmetric. So, for instance, in your case, it's whatever condition, but do you think it could be also depending on the cytoplasmic conditions, let's say?
I think so, might be. I do not really, I mean, it's more an intuition, but there's another thing that we observed in at least like by measuring the dynamics of the microtubules, is that at the beginning of the process, microtubules, as soon as you beam them with blue light, microtubules grow in the unbounded growth regime state, which is something that we did not expect, because initially we have a transverse array which is at the equilibrium, which means that the amount of available tubulin should be balanced with the number of dynamic microtubules in the transverse direction. So adding new microtubules in the longitudinal direction, it is possible, but I would expect that these microtubules are also in a bounded growth regime state, which is not what we observe. Initially, microtubules are in the unbounded growth regime. So it might very well be that in the moment in which you beam the cell with blue light, you can have something that happens in the cytoplasm. Yeah, so that's a possibility.
That's good. Yeah, we have plenty of your work is opening many other people, so that's great. Maybe I have one last very open question. So because as like something that is relevant for the journal, what would be, so as a modeler, what would be the advice you would give to other modelers who want to work on experimental data?
Okay, I think that the first advice is to do completely different from what I did at the very beginning, because at the very beginning, as a physicist who joined a very specific biological mechanism, like a project on a very specific mechanism. Initially, I have to say I was not very much interested in the biological properties of the system. I was more absorbed by my model. Mm-hmm. So I spent the few months, like the first few months, maybe the first year, only caring about my equations and caring about the design of my model. And of course, I basically did not achieve anything. I started achieving something in the moment in which I understood that modelers and experimentalists have to communicate and have to find a common ground. So my advice is listen to what the experimentalists say, because 99% of the cases, something they have to say something smart or something that can help you in making a better model or making a model that has some kind of biological relevance.
Very nice. Okay, that's a inspiring point. All right, so well, thank you very much for your talk. So I think I'll hand it over to Myra for the final words.